configuration of protein or protein structure. There are 4 levels of protein structure. One is primary, secondary, tertiary and quaternary structures. So first coming to the primary structure. The primary structure of protein is defined as the linear sequence of amino acid residues making up its polypeptide chain. The protein may be formed of one or more polypeptide chains. The amino acids are arranged in a specific sequence in the polypeptide chain. The amino acid residues are linked by peptide bonds. The peptide bond is formed between the carboxyl group of one amino acid and the amino group of adjacent amino acid. Sometimes the adjacent polypeptide chains are linked by disulfide bond. Many structural proteins which form fibers are linear and unfold types, example silk fibroin. The peptide bond is a repeating unit. 2 amino acids giving a dipeptide, 3 amino acids give a tripeptide, 4 amino acids give a tetrapeptide, and so on. Less than 10 amino acids from an oligopeptide. More than 10 amino acids form a polypeptide. Each polypeptide chain of any length has at one end an N-terminal amino acid containing a free amino group and at the other end a C-terminal amino acid containing a free carboxyl group. The amino acids in the polypeptide chains are numbered from N-terminal end. Thus, the primary structure has following silent features. That is, one is primary structure refers to the linear sequence of amino acid residues. The proteins are linear and unfolded. The protein is formed of one or more polypeptide chains. The amino acid residues are linked by repeating peptide bonds. The adjacent polypeptide chains are linked by disulfide bonds. Most of the structural proteins which are in the form of fibers exhibit primary structures. Example, fibroin of silk. Next, coming to the secondary structure. Secondary structure refers to the helical nature of the proteins. The secondary structure is derived from the primary structure by the formation of hydrogen bond interactions between amino acid residues fairly close to one another. This leads to the folding of the polypeptide chain into a helix. The intramolecular hydrogen bonds occur between an amine nitrogen and the carboxyl oxygen of the 4th amino acid residue of the polypeptide chain. In addition, disulfide bond also occur either in the same chain or between polypeptide chains. By configuration, the secondary structure is classified into 2 types, namely helical structure and plated sheets. The helical structure. The polypeptide chain is coiled as a rope wound around a cylinder. The helical structure of protein is formed by the hydrogen bond between peptide groups within the same polypeptide chain. The most common from the coiling is straight-handed alpha helix. Only right-handed alpha helix occur in nature. The alpha helical structural model was proposed by Pauling and Corey. The main features of alpha helix are: the helix is formed by a series of amino acid residues woven into a spiral chain. The alpha helix is so named because of the mobility of alpha carbon atoms. The helical chain has a screw-type symmetry. A hydrogen bond is formed between the peptide groups, that is C double bond O and NH, of every first and fourth amino acid residues. The regular appearance of hydrogen bonds between every first and fourth peptide groups determines the regularity of turns in the polypeptide chain. The shape of the helix is maintained by hydrogen bonds. Each complete coil contains 3.6 amino acid residues. These are about the alpha helix. Next, coming to the beta plated sheets, the structure of beta structure. It is a secondary structure formed from hydrogen bonding between 2 peptide chains. This bonding leads to the formation of sheets of parallel chain in the form of pleated sheets. In pleated sheet structure, the chains may be parallel or anti-parallel. In a parallel chain pleated sheet structure, the N-terminal end of the polypeptide chains points in the same direction. Example, keratin. In antiparallel chain, the pleated sheet structures, the N-terminal ends of the polypeptide chain points is opposite directions. Example, fibroin of silk. Next one is tertiary structure. The tertiary structure of protein is more complex than the secondary structure. Tertiary structure is exhibited by proteins having only one polypeptide chain. The tertiary structure is attained by globular proteins. In such case, the secondary helical structure is further folded to form a globular or ellipsoidal shape. The folding is established by the appearance of more disulfide bonds as well as hydrogen bonds, ionic bonds and hydrophobic bonds. The myoglobin, ribonuclease, chymotrypsin, cytochrome C, etc. is exit in tertiary structure. Myoglobin is abundant in muscles. It contains a single polypeptide chain of 153 amino acids and a single iron porphyrin, that is heme group. The tertiary structure of myoglobin has a Sages-like outline. The backbone of the molecule is made up of 8 almost straight helical segments, alternated by non-helical bends, which twist to produce a characteristic folding. The longest helical segment has 23 amino acids and the shortest segments has 7 amino acids. All the helical segments are of alpha type and right-handed. There being no beta structure. Nearly 80% of amino acids remain on the helical regions and the 20% are distributed over the non-helical areas. The next one, ribonuclease, is another globular protein with tertiary structure. It is made up of a single polypeptide chain containing 124 amino acids with 4 disulfide bonds. The final one is quaternary structure. 2 or more polypeptide chains associated together to produce a quaternary structure. It is exhibited by proteins containing more than one polypeptide chains. Quaternary structure is formed by the combination of primary, secondary and tertiary structure. The quaternary structure presents itself as an aggregation of 2 or more polypeptide chains with a tertiary structure organized into a single function protein molecule. The protein with a quaternary structure is referred to as an oligomer and its polypeptide chain with tertiary structure are referred to as protomers, that is protomers. The protein insulin is made up of 2 polypeptide chains. Hemoglobin has 4 polypeptide chains. Apoprotein has 24 identical chains. Aspartate transcarboxylase has 72 chains. The quaternary structure is of 2 types depending on the nature of polypeptide chains. They are homogeneous quaternary structure and heterogeneous quaternary structure. The homogeneous quaternary structure, in this quaternary structure, the polypeptide chains are identical. Example, phosphorylase, lactic acid dehydrogenase, isoenzymes H4 and M4. The second one is heterogeneous quaternary structure. In this quaternary structure, the polypeptide chains are non-identical. Example, globulin, keratin. The quaternary structure is maintained by globular and fibrous proteins. For example, hemoglobin is a globular protein and keratin is a fibrous protein, and both of them exhibit quaternary structure. The quaternary structure is maintained by hydrogen bond, disulfide bonds, hydrophobic bonds and ionic bonds. The same bonds involve the formation of tertiary structure of protein. Hemoglobin. Hemoglobin contains 4 identical polypeptide chains, 2 identical alpha chain and 2 identical beta chains. Each chain is characterized by its own secondary and tertiary structure. Each chain has heme prosthetic group. The alpha chain has 141 amino acid residues and beta chain has 146 amino acid residues. The protein has 574 amino acid residues. The alpha and beta chains have several segments of beta helix separated by bends. The alpha and beta chains are held together as a pair by ionic and hydrogen bonds. These 2 pairs of chains are joined each other by additional ionic bonds, hydrogen bonds and hydrophobic bonds. Thus, 4 polypeptide chains fit together to produce the quaternary structure. Structure of proteins. Proteins are polymers of amino acid monomers. The amino acids are linked together to form a chain called peptide chain. The peptide chain has variable length and it depends on the number of amino acids linked. The amino acids of proteins are called residues. The peptide chain is named according to the number of amino acid residues. Dipeptide. A dipeptide contains 2 amino acid residues. Tripeptide. A tripeptide chain is formed of 3 amino acids. Oligopeptide. An oligopeptide is made up of less than 10 amino acids. The final one is polypeptide. A polypeptide chain is made up of more than 10 amino acids. Proteins are polypeptides with more than 100 amino acid residues. A protein is formed of only one amino acid chain or more than one polypeptide chains make up a protein. Example, myoglobin is made up of single polypeptide chain, but hemoglobin is made up of 4 polypeptide chains. N and C terminals. The polypeptide chain of protein has directions. It has 2 definite ends. At one end, a free amino group is present. This end is called as amino terminal or N-terminal end. And the amino acid bearing free amino group is called N-terminal amino acid. At the other end, a free carboxyl group, COH group is present. This end is called carboxylic end or C-terminal end. And the amino acids bearing free carboxyl group is called as C-terminal amino acids. Chemical bonds involved in protein structure. Proteins are the polymers of amino acid monomers. Any 2 amino acid monomers are linked together by a chemical bond. There are 5 types of bonds which occurs in proteins. They are peptide bond, disulfide bond, hydrogen bond, non-polar or hydrophobic bond, and final one is ionic bond. Peptide bond. Peptide bond is an amide bond where the CO group of COH group of one amino acid is linked with NH group of NH2 group of the adjacent amino acid. The peptide bond produces the linear and primary structure of protein. Second one is disulfide bond. Disulfide bond is a covalent bridge formed between 2 polypeptide chains by a cysteine residue. The disulfide bond is formed by the oxidation of thiol, that is SH groups of 2 cysteine molecules. This results in the formation of molecule of cysteine, an amino acid with a disulfide bridge. Example, oxytocin, a hormone of pituitary gland, has a disulfide bond. Here the disulfide bond links 2 cysteine units of the same polypeptide chain. Insulin is another important for disulfide bond. In insulin, 2 polypeptide chains are linked together by 2 disulfide bonds. Another disulfide bond connects with amino acid 6 and 11 in A chain. The disulfide bond produces a linear form and gives the primary structure of protein. Third one is hydrogen bond. Hydrogen bond is a weak electrostatic attraction between one electronegative atom and a hydrogen atom covalently linked to a second electronegative atom. The formation of a hydrogen bond is due to the tendency of hydrogen atom to share electrons with 2 neighboring atoms, especially oxygen and hydrogen. For example, the carboxylic oxygen of one peptide bond shares its electrons with the hydrogen atom of another peptide. An interaction sits in between a C double bond O group and the proton of an NH or OH group. If these groups come within a distance of about 2.8 angstrom, this secondary valency bond is symbolized by dotted, since that is C NH. Silk fibrin is an example of the presence of hydrogen bond involving the imide and carbonyl groups of the peptide bond. Here the hydrogen bond links the vaginal peptide chains. In keratin of full, the hydrogen bonds link the side chains so that the single peptide chain is held in a coiled or helical forms. Hydrogen bonds maintain the helical nature and produce secondary structure of proteins. Fourth one is non-polar or hydrophobic bond. The association of non-polar groups with each other in aqueous system because of the tendency of the surrounding water molecules to seek their most stable state is called nonpolar or hydrophobic bond. Many amino acids like alanine, valine, leucine, isoleucine, methionine, tryptophan, phenylalanine and tyrosine have hydrophobic side chains. That is, they have little attraction for water molecules in comparison to the strong hydrogen bonding between water molecules. Such R groups can unite among themselves with the elimination of water to form linkages between various segments of a chain or between different chains. This is very much like a oil droplets suspended in water. The fifth one is ionic or electrostatic bond. Ionic bonds are formed by ionization. The bond is formed by the transfer of electrons, that is ions, from one atom to another. Ions possessing similar charge repel each other, whereas the ions having dissimilar charge attract each other. For example, divalent cations like magnesium may form electrostatic bonds with two acidic side chains. Another instance of ionic bonding may be the interaction between the acidic and basic groups of the constituent of amino acids. Ionic bonds occur between positively charged groups and negatively charged groups. The ionic bonds maintain a folded nature of proteins and produce tertiary structure.